humans. Types of blood group in human and some problems based on the blood group. Here, if the father is homozygous A blood group and mother blood group is homozygous B, what is the probability of the children blood group is? How to determine the blood group? Here, if the father is A, the father blood group is A and mother blood group is B. So condition is homozygous. Homozygous A blood group, father blood group, and homozygous B blood group, mother blood group. This is mother blood group and this is father blood group. So what is the condition of blood group in the children? Here the genotype of A blood group. I already told the blood group A consisting of the genotype IA. In heterozygous condition I. Here B blood group IB, and in heterozygous condition IB. AB blood group IA, IB, and O blood group I. So these are the genotypes of the blood group. Here condition is father blood group is homozygous. So genotype is IA. Mother blood group is B. The condition of the genotype is IB. So how many gametes are produced in this alleles? Only one type of gamete is produced because the condition is homozygous. IA and IA is same. The condition is homozygous. That's why we represent it as a gamete 1. And this is also IB because condition is homozygous. So crossing is takes place IA, IB. So IA, IB is a genotype, blood group AB. So the children may contain the blood group AB. Likewise, I given some problem and solve those problem. I will check in regular class. Okay. Next concept is about Mendel's work on principles of inheritance is unrecognized. Mendel ಏನು ವರ್ಕ್ ಮಾಡಿರ್ತಾನಲ್ರಿ ಮೊನೋಹೈಬ್ರಿಡ್ ಡೈಹೈಬ್ರಿಡ್ ವರ್ಕ್ ಅನ್ರಿಕಗ್ನೈಸ್ ಆಗ್ತದ ಸೊಸೈಟಿ ಒಳಗ ಅದನ್ನ ಗುರ್ತು ಹಿಡಿಯಂಗಿಲ್ಲ. Why? Because in Mendel's time the communication is very low. Communication is not good. This is first reason. That's why Mendel's work is unrecognized. And second reason, Mendel's used the mathematics in his experiment. That mathematics is unacceptable to many biologists because in that time the biologists couldn't understand about mathematics in the biology. That's why the work is unrecognized. So he used meiosis. During meiosis. This is first one. Second is about the behavior of the chromosomes, always parallel to the behavior of the genes. Behavior of chromosomes parallel to behavior of genes. Third point, Sudan united the knowledge of chromosomal theory of inheritance or chromosomes theory and Mendel's principle. He united chromosomes theory plus Mendel's theory. So, and gives the name of chromosomal theory of inheritance. Next is about important key points about chromosomal theory of inheritance. Here, in this point, the chromosomes are containing the many genes. First point, some important points regarding to chromosomal theory of inheritance. Here, the chromosomes contain many genes. One chromosome ಒಳಗ many genes present ಇರ್ತಾವ. Second character, the genes are always pair. Those are called as alleles. And genes are occur at a place that is called as a locus. That is nothing but a place of a genes. That is called as a locus. This is about some key points. Chromosomes are containing the many genes. Those genes are always in the pair. Those are called as a alleles. And alleles are present on the some place on the chromosomes. That is called as a locus. This is about some key points about chromosomal theory of inheritance. Here, next is about chromosomal theory was experimentally proved by. Next is about chromosomal theory. Chromosomal theory was experimentally proved by T.H. Morgan. ಇಲ್ಲಿ ಏನಾಗ್ತದ ರೀ, ವಾಲ್ಟರ್ ಅಂಡ್ ಸುಟಾನ್ experimentally proof ಕೊಡಂಗಿಲ್ಲ. He explained only about theory, no experiment. Here the T.H. Morgan explained the chromosomal theory of inheritance by experimentally. ಇವ್ ಮೆಂಡಲ್ ಹೆಂಗ್ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡ್ತಾನಲ್ಲ monohybrid and dihybrid cross ಒಳಗ, so likewise the T.H. Morgan Morgan. T.H. Morgan is nothing but a Thomas Hunt Morgan. Full name of T.H. Morgan. Why he used a Drosophila for his experiment? Mendel pea plant ಅನ್ನು select ಮಾಡ್ಕೊತಾನ್ರಿ experiment plant ಆಗಿ because the plant containing the some characters. ನಿಮಗೆ ಗೊತ್ತದರಿ ಯಾವ ಕ್ಯಾರೆಕ್ಟರ್ ಅದ ಅಂತ ಹೇಳಿ ಪ್ಲಾಂಟ್ಸ್ ಒಳಗ ಕೆಲವು ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಅದಾವ. that's why pea plant ಅನ್ನ select ಮಾಡ್ಕೊತಾನ. ಸೊ ಅದೇ ರೀತಿ ಏನ್ ಮಾಡ್ತಾನ್ರಿ T.H. Morgan ನ ಅವನ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡಾಕ ಡ್ರೋಸೋಫಿಲಾ ಅನ್ನ ಸೆಲೆಕ್ಟ್ ಮಾಡ್ಕೊತಾನ. why? because there are some characters are present in this fruit plant. This is also very important. This asked for a 2 marks question. It is 2 mark question. Why the T.H. Morgan select the Drosophila for his experiment? Because first reason, the Drosophila grow in synthetic media. It will grow in synthetic media. That is in the laboratory condition. In laboratory condition or in lab. So synthetic media is nothing but a artificial media. Artificial media. Artificial media is nothing but a nutrient media. For example, I told in first year, agar is a synthetic media used for a growing of the bacteria. Likewise, the Drosophila is grow easily in a laboratory condition in the synthetic media. That is first reason. Second reason, the Drosophila consisting of the minimum number of chromosomes, that is 2n equal to 8. The number of chromosomes are very small. That help in the studying of the behavior of the chromosomes. Third reason is the life cycle of Drosophila is short. Life cycle of fruit fly is short. Within 15 days, that is 15 days. Within 15 days, the Drosophila complete their life cycle. This is third reason. Fourth reason, it show sexual dimorphism. It will show sexual dimorphism. The sexual dimorphism is nothing but a clear difference between male and female fruit fly. Write up in bracket, write male and female. Clear difference between male and female flies. That is called as a sexual dimorphism. That is fourth reason. Here fifth reason is the chromosomal behavior of the fruit fly can be easily observed under a microscope. The behavior of chromosomes easily observed under microscope. So these are the some reasons for selecting the Drosophila. These are all characters are present in the fruit fly, that is Drosophila. That's why T.H. Morgan select the Drosophila for his experiment. complete linkage and incomplete linkage. Here complete linkage. Consider this is a chromosome. Chromosome. If the two genes are close to each other, ಇವೆರಡು genes ಇದಾವೆ, ಎರಡು close ಇದಾವ. Close to each other. Distance ಏನದಾರಿ? Short ಅದ. So that is called as a complete linkage. So ಏನಾಗ್ತದರಿ? ಇವು close ಇರ್ತಾವ. These genes are remains close to each other from generation to generation. That is called as a complete linkage. In incomplete linkage, consider this is a chromosome. The genes are apart from each other. Genes distance ಏನದರಿ? ದೂರದ. Distance is long. Those genes are same together from generation to generation. Those are called as a incomplete linkage. Incomplete genes. The here genes are together, the genes are apart from each other. Second is about incomplete linkage. The complete linkage, there is no crossing over of the genes. ಏನಾಗ್ತದೆ complete linkage ಒಳಗ genes are close ಇರಲ್ಲ, crossing over ಆಗಂಗಿಲ್ಲ. Crossing over means the genes are gets crossed each other. There is no crossing over in the complete linkage. So that leads to there is no variation in the organism. Here incomplete. The crossing is possibility. Genes ದೂರ ಇರಾನೆ ಏನಾಗಿರ್ತದರಿ? Crossing ಇರ್ತದ. Crossing over is possibility. Crossing over. Here no crossing over. No crossing over of the genes. Here crossing over of the genes. So crossing over ಆಗನೆ ಏನಾಗ್ತದರಿ? genes ಒಳಗೆ crossing over ಆಗನೆ ಏನಾಗ್ತದರಿ? variation ಆಗ್ತದ. So variation that leads to the evolution of organism. So ಏನಾಗ್ತದರಿ? changes genes ಒಳಗೆ variation ಆಗೋದ್ರಿಂದ ಏನಾಗ್ತದರಿ? evolution ಆಗ್ತದ. So this is about types of linkage, complete and incomplete linkage. So next is about the T.H. Morgan. I told he select the fruit fly for his experiment and he made a two crosses and give the name as a cross A and cross B. Because to conclude about linkage and recombination. ಇವ ಏನ್ ಮಾಡ್ತಾನ್ರಿ? linkage and recombination ಅನ್ನ ಸ್ಟಡಿ ಮಾಡಕ್ ಏನಾಗ್ತದ? ಕ್ರಾಸ್ ಮಾಡ್ತಾನ. cross A and cross B. So cross A ಒಳಗೆ ಏನ್ ಮಾಡ್ತಾನ್ರಿ? ಡ್ರೋಸೋಫಿಲಾ ದ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅನ್ನ ಏನ್ ಮಾಡ್ತಾನ್ರಿ ಅವ? ತಗೋತಾನ ಅಥವಾ ಅಬ್ಸರ್ವ್ ಮಾಡ್ತಾನ. So those character cross A ಒಳಗೆ ಯಾವ ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ಅನ್ನ ಕ್ರಾಸ್ ಮಾಡ್ತಾನ ಅಂದ? First character is about brown body, red eyes. One ಡ್ರೋಸೋಫಿಲಾ ಅದರಿ. Consider this is a fruit fly. Fruit fly. 98.7% of Drosophila became wild type. Wild type. That is brown body, red eye. But 1.3% is became mutant trait. Mutant trait ಅಂದ್ರೆ ಏನಾಗಿರ್ತದೆ ರೀ? Here brown body, white eyes. Mutant trait ನೋಡಿ, wild type ಒಳಗೆ ಏನದ ರೀ? brown body, red eyes ಅದಾವ. But in mutant trait, brown body, white eyes and yellow body, red eyes. This is important. Mutant trait. 1.3% is mutant trait. This is the result of first cross. ಏನ್ ಮಾಡ್ತಾನೆ ಫಸ್ಟ್ ಕ್ರಾಸ್? ಮೇಲ ಏನಾಗ್ತದ ರೀ? ಬ್ರೌನ್ ಬಾಡಿ ಇರ್ತದ, ರೆಡ್ ಐಸ್ ಇರ್ತಾವ. ಅವನು ಎದ್ರ ಜೊತೆ ಕ್ರಾಸ್ ಮಾಡ್ತಾನ್ರಿ? ಫಿಮೇಲ್ಸ್ ಏನಾಗಿರ್ತಾವ್ರಿ? ಯೆಲ್ಲೋ ಬಾಡಿ ಇರ್ತದ, ವೈಟ್ ಐಸ್ ಇರ್ತಾವ. ಸೊ ಕ್ರಾಸ್ ಮಾಡಿದಾಗ F1 ಜನರೇಷನ್ ಒಳಗೆ ಏನಾಗ್ತದ ರೀ? ಆಲ್ ಫಿಮೇಲ್ಸ್ ಬಿಕೇಮ್ ಏನಾಗ್ತದ ರೀ? ಫಿಮೇಲ್ಸ್ ಅಪೋಸಿಟ್ ಅಂದ್ರೆ ಮೇಲ್ ಕ್ಯಾರೆಕ್ಟರ್ ಬಂದ್ಬಿಟ್ಟಿರ್ತಾವ್ರಿ. ದಟ್ ಈಸ್ ಬ್ರೌನ್ ಬಾಡಿ, ರೆಡ್ ಐಸ್. ಅಂಡ್ ಮೇಲ್ಸ್ ಏನಾಗಿರ್ತಾರೆ? ಯೆಲ್ಲೋ ಬಾಡಿ, ವೈಟ್ ಐ ಆಗಿರ್ತಾವ. ಅವು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ಅಗೇನ್ F1 ಜನರೇಷನ್ ಒಳಗೆ ಮೇಲ್ಸ್ ಫಿಮೇಲ್ಸ್ ಏನ್ ಬಂದಿರ್ತಾವಲ್ಲ, ಅವು ಕ್ರಾಸ್ ಮಾಡಿದಾಗ ನೆಕ್ಸ್ಟ್ ಜನರೇಷನ್ ಏನ್ ಬಂದಿರ್ತದ ರೀ? 98.7% ಆಫ್ ವೈಲ್ಡ್ ಟೈಪ್. ದಟ್ ಈಸ್ ಬ್ರೌನ್ ಬಾಡಿ ಅಂಡ್ ರೆಡ್ ಐಸ್. ಅಂಡ್ 1.3% ಆಫ್ mutant trait. ಏನಾಗಿರ್ತದೆ ರೀ mutant trait ಒಳಗ? ಬ್ರೌನ್ ಬಾಡಿ, white eyes and yellow body, red eyes. That is mutant trait. Here next is about cross B. Cross B ಏನ್ ಮಾಡ್ತಾನ? Here in a cross B he took about wings. Shape are normal wings and miniature wing. Miniature is nothing but consider this is a Drosophila. The miniature means small wings. And normal wings is nothing but the Drosophila containing a normal wings. The characters are crossed. White eyes, miniature wing. This is mutant trait. Those are females. Females ಏನಿರ್ತಾವ್ರಿ? White eyes. Eyes ಏನಿರ್ತಾವ್ರಿ? White ಇರ್ತಾವ. Wings ಏನಿರ್ತಾವ್ರಿ? Small ಇರ್ತಾವ. So these Drosophila are crossed with a normal wild type. Is nothing but a red eyes. Red eyes, normal wings. This is cross B. Parents of cross B. Here, in this cross, 68.2% are wild type. That is wild type means this one. Normal wild type means red eyes, normal wing. ಎಷ್ಟು ಪರ್ಸೆಂಟ್ ಇರ್ತಾವ್ರಿ? 62.82% ಇರ್ತಾವ. So next about 68.2%. Next about 28.6% is mutant trait. That is mutant trait ಅಂದ್ರೆ ಏನ್ರಿ? ರೆಡ್ ಐಸ್ ಇರ್ತಾವ, ಬಟ್ ವಿಂಗ್ಸ್ ಏನಿರ್ತಾವ್ರಿ? ಮಿನಿಯೇಚರ್ ಇರ್ತಾವ. ಆಮೇಲೆ ನೆಕ್ಸ್ಟ್ ಏನಿರ್ತಾವ್ರಿ? ವೈಟ್ ಐಸ್ ಇರ್ತಾವ, ಬಟ್ ವಿಂಗ್ಸ್ ಏನಿರ್ತಾವ್ರಿ? ನಾರ್ಮಲ್ ಇರ್ತಾವ. That is mutant trait. That's why from these two cross, the Morgan conclude that linkage, if the linkage, conclusion of this experiment is if the linkage is more, the recombination is less. If the linkage is less, the recombination is more. That is conclusion. ಇದರೊಳಗೆ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಒಳಗೆ ಏನು ಕೇಳ್ತಾರೆ? ಮೋಸ್ಟ್ ಆಫ್ ದ ಕ್ವೆಶ್ಚನ್ಸ್ ಏನು ಬಂದಾರೆ? ರೈಟ್ ದ ಕನ್ಕ್ಲೂಷನ್ ಆಫ್ ಡ್ರೋಸೋಫಿಲಾ ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್. So that is conclusion is very important. Here conclusion conclusion of the experiment. Right. Conclusion. If linkage is more, recombination is less. If linkage is less, linkage is less, recombination is more. This is important. 2 marks. Conclusion of this experiment. In cross B, I will mention 62.8 in 62.2, that is wrong. So 62.8 and 37.2%. ಏನಂತಾರೆ ಕ್ರಾಸ್ ಬಿ ಮಾಡಿದಾಗ 62.8% ಪೇರೆಂಟಲ್ ಟೈಪ್ ಇರ್ತಾವ. ದಟ್ ಈಸ್ ಪೇರೆಂಟಲ್ ಟೈಪ್ ಅಂದ್ರೆ ಏನ್ರಿ? ನಾರ್ಮಲ್ ರೆಡ್ ಐಸ್ ಅಂಡ್ ನಾರ್ಮಲ್ ವಿಂಗ್ಸ್. ರೀಕಾಂಬಿನೇಷನ್ ಟೈಪ್ ಅಂಡ್ ಮ್ಯೂಟೆಂಟ್ ಟ್ರೇಟ್, ರೀಕಾಂಬಿನೇಷನ್ ಟ್ರೇಟ್ ಏನಿರ್ತದರಿ 32.7% is ಏನಿರ್ತದರಿ ರೆಡ್ ಐಸ್ ಸ್ಮಾಲ್ ವಿಂಗ್ಸ್ and white eyes larger wings. That is this percentage is red. So next is about sex determination in individual. How the sex is determined by the individuals. Here the XY chromosomes are present in the individuals. So X and Y chromosomes are responsible for a determination of the sex of an individual. So Henking, it is the name of a scientist. Henking observed the movement of the chromosomes during the spermatogenesis, or he observed the some unacceptable chromosome in the during the spermatogenesis. Those determination mechanism. Male heterogamety and female heterogamety. Here what happened? Male heterogamety is nothing but a XX and XY mechanism. ಏನಾಗ್ತದರಿ ಮೇಲ್ ಹೆಟೆರೋಗೆಮಿಟಿ ಒಳಗ ಮೇಲ್ ಪೇರೆಂಟ್ ಒಳಗ ಏನಿರ್ತದರಿ ಹೆಟೆರೋಗೆಮಿಟಿ ನೇಚರ್ ಇರ್ತದ, ಹೆಟೆರೋಜೈಗಸ್ ಕಂಡೀಷನ್ ಇರ್ತದರಿ ಮೇಲ್ ಪೇರೆಂಟ್ ಒಳಗ. And in female heterogamety the female parent containing the heterozygous condition, female ಏನಿರ್ತದರಿ heterozygous condition present ಇರ್ತದ. So that's why male heterogamety name given as a male heterogamety is crossed with male AA plus X0. This is male parent. Here what happen? In males, the XX and X0 mechanism, the male parent loss or do not contain its partner, that is Y chromosome. Y chromosome present irangili. So in example, ಈ mechanism ಎಲ್ಲಿ present ಇರ್ತದ? So grasshopper. Grasshopper is an example of XX and X0 mechanism. ಅಲ್ಲಿ Y chromosome present ಇರಂಗಿಲ್ಲ. ನಿಲ್ಲಿರ್ತದರಿ X0 Y chromosome present ಇರಂಗಿಲ್ಲ. So here gametes A plus X. Here A plus X and A plus 0. Male parent. The crossing is done. AA plus XX. Here AA plus X0. Crossing is done. Here it is female and it is male. That's why here also male is responsible for a sex determination of an grasshopper, whether it is male or whether it is female. It is about first male heterogamity.